This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 43, recorded on August 22nd, 2012. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Good morning, Vincent.
Good morning. It's 11:41.
It is.
Eastern in New York City.
True.
Nice day out.
Not bad.
A lot of big puffy clouds out there.
Yep. Kind of reminds you of a
Temperature-
Georgia O'Keeffe painting in her
No,
latter
it
years.
does not.
It does me.
Well, that's totally out of line. It's not anything like Georgia O'Keeffe. She painted flowers, didn't she?
She painted clouds.
26 degrees
26
Celsius.
degrees. That's
What's that?
middle 80s.
We don't care.
We don't care.
We should think in Celsius.
That's true.
How have you been, Dixon?
I've been fine, Vince.
Now, the last time we recorded was less than 2 weeks ago.
It was.
We're trying to... What?
I made the sign of victory. We're trying to record every 2 weeks now. We are.
Well, we'll try.
Which should work out pretty
Probably.
well. It should work out pretty well,
I
Vince.
don't know. You and I go here and there.
We do. We do. When's your next trip?
Friday.
Where?
Cold Spring Harbor Laboratory.
Nice. And what is the occasion?
The American Society for Microbiology is commemorating Cold Spring Harbor Lab with a milestone in microbiology plaque
Fantastic.
because of its contribution to microbiology research and education over the years. And
Wonderful.
it just so happens that this week is the phage, bacteria, and archaea course there.
Oh, nice.
And so they're having a little ceremony on Friday to put this plaque, and we will be doing a TWiP out there.
That's fantastic.
Do you know what TWiP is?
I do. This
TWiP?
week in microbiology.
Yeah, so that should be fun.
Yes.
It'll be up next week. But
Yes.
this episode of TWiP,
Yes.
43,
Yes.
will go up. What's today? Wednesday?
Yeah.
I'll put it up tomorrow, which is
Wow.
Thursday.
That's fast forward.
Because
That's fast track.
if you don't breathe,
I'm going to try not to breathe, Vince, but you won't hear much from me then.
am I a pain in your side, a thorn in your side?
Not a thorn in my side. No, of
So
course
today
not.
we decided we would do a couple of research papers, right?
Correct. That's right.
Is that a good idea?
It's a great idea.
One of them was sent by a listener, and the other one you picked.
I did.
And,
Picked it right off the cover of Science magazine.
You just look at the cover. You assume that whatever's on the cover is good.
Not always.
How often does a parasite story get on the cover of Science?
It's interesting. Not often, but when it does, it's usually a blockbuster. And this particular cover of Science had a great scanning electron microscopy view of the bloodstream. And it had a number of host cells.
A blood vessel, is
Yeah,
that right?
blood vessels,
Capillary or
because
something?
trypanosomes only live in the blood vessels. Trypanosomes, in this case, the Trypanosoma brucei. It's a strictly blood vessel dwelling organism.
Some are intracellular, right?
Not Trypanosoma brucei.
Not brucei, but other trips
Trypanosoma cruzi.
that go into cells,
They
right?
do. That's right. Those are the two kinds. One's in Africa and
Yeah,
one's...
we call brucei a free living form, as if life was free.
I don't call that a free living form. It's an obligate parasite.
It's got to be in you,
It has to be in you.
but it's an extracellular.
It's
Yeah,
extracellular.
we probably discussed all of this.
We did.
So your paper, you want to do your paper first?
Sure.
Since we started talking
It's
about
not my paper, but it's the paper I selected.
it. You would like it to be.
I wish.
From Science,
No,
adenylate cyclases of Trypanosoma brucei.
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 Well, Bruce was the guy's name.
Bruce who?
That was his last name.
Inhibit the innate immune response of the host.
Yes.
And the
So innate immune
this group
responses
is
of the
from
host.
Belgium.
They are.
What's an adenylate cyclase, Dickson?
Well,
Nah.
wait a minute. No, it's a transmembrane protein,
Yeah.
and it's found throughout nature. And it's an enzyme.
Is it in you?
Of course.
And what does it make?
Well, it cyclizes ATP.
And what's the product called?
Cyclic AMP.
Wow, you know this. I was trying to trip you up.
That was a pun. Thank you.
And what does cyclic
Well, I
AMP do?
reached out with my flagella and wrapped it around your neck.
What does cyclic AMP do?
Oh, lots of different things. It regulates
if you
metabolism.
answered that on a test, I would fail you. You got to be specific.
It regulates metabolism at many levels.
And what is the name for this molecule, a general name?
Cyclases.
No. Cyclic AMP is a second messenger. Right?
Oh, you mean of the cyclic AMP?
Yeah,
The second
involved
messenger.
in
Of course.
signal transduction pathways.
Okay, name another second messenger.
Messenger RNA.
Mercury, he was a fantastic messenger. No, calcium. Calcium. It's a second messenger.
Calcifer. Okay.
Yeah.
So this paper is telling us in the title that the enzymes that make cyclic AMP
for the trypanosome
in trypanosomes inhibit the innate immune response of the host.
Right. So
what's the innate immune response?
Exactly what I wanted to lead into, Vince, because I think this opens up an enormous category for discussion. All right? Innate immunity. What does that mean?
It's there all the time.
Correct. It's not inducible.
It doesn't need, well, to a certain extent, it's always there, but there are aspects that are inducible, but it doesn't need to adapt to the invader. Right?
It's not specific.
It's not specific. That's right.
So
an antibody would be an adaptive response that's tailored to the pathogen.
That's right.
But the innate response is early, very early on. It's always active to protect you.
Yes.
And it's not specific.
So what are those components of the innate immune system?
So some of the components are molecular recognition molecules like toll-like receptors and cytoplasmic helicases that recognize microbial patterns, sentinel cells, dendritic cells, and macrophages, and NK cells. Those are the main components. Even complementary.
Natural killers.
Natural killers.
Guns N' Roses or something
So right
like
now,
that.
you and I have an active innate response. It's just checking all the time to make sure everything is us. If it detects something foreign, let's say a virus goes into your respiratory tract and it begins to replicate, it starts to make nucleic acids, and those can be sensed by sensors of the innate response. And then you respond by making cytokines. You
do.
The cytokines recruit the sentinels and get them in the area.
You do.
You make interferons.
Are there other aspects of the innate immune response that don't involve molecular biology?
Everything involves molecular biology.
Well, what about the barriers that are created?
Well, that's a intrinsic response.
First line of defense.
Yeah, but it's not innate. It's intrinsic.
Yeah, but it's innate.
It's all, it's all terminology. It's
I know.
What we call things.
Okay, what about
nature? Doesn't care what we call things.
That's true.
Right? But you're right. There's the skin. There's the mucus. There's pH. There are many intrinsic things. What else?
There are peptides that are
meganins.
Yes, that's right. And defensins. All of those molecules fall into that category as well.
So in my textbook, we actually call that intrinsic.
Really?
Because it's not immune.
Well, it seems to ward off the evil
No,
spirits.
but it's all terminology. It doesn't really matter.
No, it doesn't. But
if pathogens breach these non-specific defenses, skin, mucus, low pH, then they encounter the innate defense. And most infections are stopped
at the molecular level
by the innate response. And if it gets over that, then it encounters the adaptive response, which takes about 2 weeks to mature.
Right.
And if it gets over that, you're dead.
Or you become balanced polymorphism.
I'm not a polymorphism.
No. So I knew her, by the way. I used to date her in school. Yes, that's right.
Every night?
I'm here every night.
Try the veal.
I think the veal is not bad. So when you see the title of an article like this, you're bound and determined to think of the innate immune response in the molecular sense, right? Not in the barrier sense, because this organism is already inside the host.
Exactly, it's already gotten through the.
And how did it get there, Vince?
It was delivered by the bite of a tsetse fly.
Correct.
We should review a little trypanosome biology. So this is T. brucei, which is delivered by the tsetse fly. It's a vector, and it bites you.
Yes.
Where does the trypanosome come from in the fly? Does it defecate and put it on you, or does it come out of the mouth parts?
It comes out of the mouth parts.
So as it's biting you, it's delivering trypanosomes?
That's correct.
And it has picked up the trypanosomes from another human.
Or an animal.
It could be also from so a T. brucei is in animals?
Well, there are two kinds, remember?
I want to know about T. brucei.
Well, T. brucei has other names. It's T. brucei rhodesiense.
Ah, yes, right.
Or T. brucei gambiense.
And so where do they come from?
They come from East and West Africa.
And from humans or animals, right?
Mostly animals. It's not
what kind of animals?
Four-legged animals.
So we talked about this on a previous trip.
Yeah.
TWiP, not TRIPS. We did 2 TWiPs on trips.
We did.
Can you say that really fast?
2 TWiPs on trips. And then I took a trip, and then I had to come back.
So when a fly bites you, your skin is it didn't work. It's breached.
Right.
It didn't work, right?
That's right.
Now, if you had thick skin, maybe, but there has not been enough selective pressure on humans to make them thick-skinned.
Well, some of them, they become chairmen.
Probably there's a downside to having thick skin. I would guess you can't respire very well.
Of course.
Transpire.
Of course.
Right, so
exactly right. Exactly.
But on the other hand, there probably aren't that many places where you get bitten by tsetse flies, and hence there hasn't been pressure on the whole humans population.
Avoided this infection early on in our development by actually migrating out of Africa.
A movie called Out of Africa, wasn't it?
This is true.
It has nothing to do with trips, though, right?
Isaac Dennison was the writer of that one.
So when people got too sick in certain places, they left.
They did. They avoided them.
It's like when you get sick of your chairman, you leave.
And vice versa.
When your chairman gets sick of you,
right.
They fire you. Although if you're tenured, you can't be fired.
No, but life can be made difficult.
Yes. But getting back to trypanosomes.
Yes, please.
So they breach the intrinsic defenses.
They're injected, and then they get in the bloodstream. Eventually. Remember, because in the beginning they don't.
I don't remember.
They form these lymphatic drainage lesions, right?
Mm-hmm.
And so there's something called shagomas. A shagoma is a lesion formed by the Trypanosoma cruzi group. Mm-hmm. And usually when they defecate, the reduvid bug defecates around the eye, you rub the feces of the bug into your eye, and you get a local infection to begin with. Well, the same thing happens with Trypanosoma brucei, rhodesiense, or gambiense. When you first get bitten, you get this lesion that stays local. All right? Mm-hmm. And the organisms reproduce like crazy inside the lymphatic drainage of that particular area where you were bitten. And from that point on, then they invade the bloodstream.
How long is that period of time?
2 or 3 weeks.
Yeah.
So that's what we're talking about here. How does this organism survive long enough for 2 or 3 weeks' worth of reproduction in order to get enough organisms to invade the bloodstream? That's the question. Mm-hmm. And they're addressing this question of there must be something inhibiting some basic innate immune response, because you said that the adaptive immune responses take about 2 weeks to develop.
Yeah, that's one of the main functions of the innate response, is to delay.
Right.
Either eliminate or delay until the adaptive
That's
can
correct.
kick in.
So how do these organisms overcome that?
Those are the big guns.
Because we already know how they overcome the adaptive immune response.
Well, it's a good question, because for viruses we know that every virus genome encodes at least one protein that antagonizes innate responses.
See that?
Otherwise, they would be eliminated.
Of course.
Because the innate system is so effective.
How do we interfere with their interference? Right?
We do counter attacks.
No, I mean with a drug or something like this. How can we inhibit the organism from inhibiting ours?
If you identify a pathogen protein that's inhibiting
so that's
innate,
what this paper suggests strongly.
But sorry, go ahead.
No,
I suggest strongly.
I'm absolutely knocked out by the altruism that this organism shows for its progeny.
All right, well, we'll get to that.
Yeah.
So these trypanosomes are covered with a variant surface glycoprotein?
Not to begin with, yes, but it's when they go from the trypanosome to the host,
They
right?
make that?
They're metacyclic trypanosomes when they are injected.
Okay.
And then they become trypomastigotes in the tissue. All right, so there's this transition form, all right? They have a surface coat protein, but it's totally different than the variant antigens that you would associate with the bloodstream form of this trypanosome.
Okay.
Okay? So I think it's called PARP, P-A-R-P.
What is called PARP?
PARP, the coat that's on the
I see. Okay.
bloodstream form of the trypanosomes. But I'm sure that there are listeners that are just cringing at my lack of knowledge and will write us to tell us, no, that's not PARP.
Oh, they can write it.
The PARP is something else, but I have always been led to believe that that the protein that coats the surface of the trypanosome as it exits the tsetse fly and into us is totally different than the surface coat antigens that develop over time in sequence once they enter the bloodstream.
Okay.
So
this story begins with their observation that, unlike related trips that grow intracellularly, T. cruzi, and then they also cite leishmania, which is an intracellular guy, right?
it has a promastigote form that looks a little bit like trypomastigotes.
The extracellular trips, T. brucei,
Yes.
T. congolense,
Yes.
and
T. evansi.
Oh, they don't list that. They list T. vivax.
Okay, T. vivax in sheep.
They show a remarkable expansion of adenylate cyclases.
Interesting.
All right, the genome of T. brucei, which has been sequenced,
Yep, fully.
encodes more than 80
Oh my goodness.
adenylate cyclases.
I'll be darned.
And they say many of these are on the cell surface. So that's a lot.
It is.
And you might look at this and say, it's got to mean something.
It does.
And let's figure it out.
Yeah.
So that's what this paper is about.
Sure.
And so they don't know what the function of these genes encoding, let's call them adenylate cyclases, ACs.
Right, because
They don't know what they do.
Because if cyclic AMP is a second messenger and it's intracellular, then what's an intracellular enzyme doing on the outside of an organism?
So if you have a gene encoding something and you don't know what it does, what do you do?
Yeah, knock it out.
You could knock it out.
Or you amplify it.
You could do that too. We could overexpress it.
Yeah.
But they didn't do either of those things in this paper.
No, they didn't.
Probably for technical reasons.
Maybe.
But they don't elaborate. They made a dominant negative form.
And you're going to explain that to us, aren't you?
Okay, so adenylate cyclases are active when they're dimers.
Right.
Or dimmers, light dimmers.
Right. They have
Dimers.
alpha chains and beta chains.
The same protein, just two of them come together and make a dimer, and that is the active enzyme that can now make cyclic AMP from
AMP.
ATP.
ATP.
Yeah, that's right. I'm just making sure you're keeping up here.
I said ATP.
So when you have multimeric proteins such as this,
Yes.
dimers, trimers.
And there's a lot of examples of all that, right? The toll receptors,
Yeah,
and
tons.
antibody molecules, et cetera, et
Yeah,
cetera.
if you make in the cell a defective monomer that can now pair with a good monomer from the cell,
You'll interfere with its
that will
abilities.
interfere. So you'll have a protein made up of one good chain and one mutant chain.
So is this a dominant negative?
So it's dominant because it interferes with the function of the whole protein.
And it's negative because it
No
interferes.
activity, yeah.
Right.
So they previously identified amino acid changes, just a few, that when put in a protein, adenylate cyclase, and expressed in TRIPS, make inactive enzymes, basically.
This requires transfection ability. Yes.
Yeah, you put these genes on plasmids and you introduce them, which of course for many years was impossible or difficult, and then more recently was developed for TRIPS, and that's what allows fields to go forward.
Exactly right.
And have papers in science, otherwise you're excluded.
Right.
Because you can't do any
right. You can't do genetics.
genetics. Yeah, it's
That's
very
right.
hard.
That's right.
So they inactivated the catalytic domain, which is where all the enzymology happens. You just make a few amino acid changes in there. You don't want to muck up the protein because then it won't dimerize.
Right.
And it won't be dominant negative.
Right.
Right. So let's say I have an enzyme that's a monomer, Dickson. Can I make a dominant negative?
No.
No.
You're just seeing if I'm awake now. Is that the idea? Vince, at one time I was actually
You were smart.
conversant in this language of molecular biology, but
I'm just using
I understand.
talking as a teaching method.
Yeah. No. I will be the class dummy if you'd like, because that's an easy role for me to play anyway. So
now they made several dominant negatives. They made one with 3 or 2 amino acid changes, and then a couple with individual ones, and we'll encounter those a bit. And they put the genes into TRIPs, and they make them in a way that's inducible. They don't want constitutive expression.
Right.
So they can induce them, and we don't need to go into how they induce them. So you can put the gene into TRIPs, they're fine.
Yep.
And then you add a drug, and it induces.
Right.
And they say when they do that, the total adenylate cyclase activity that they can measure by an enzymatic assay is inhibited 50%.
Right.
Okay?
Right.
So that's okay. You could.
Yep.
That's enough that you could hope to see an effect.
Right.
And if you didn't, you'd have to go back and get better than 50%.
Right. But they're using an animal model here to test it out in, right? Because otherwise, there would be no innate immune response to measure.
Of course. There's no innate immunity in...
That's right.
Well, there is some innate immunity in cells. You can measure toll-like receptors.
Yeah,
and you can measure interferon production.
You could do that.
And the effect on, say, growth of parasites. But
That's
you really
right.
want to know in animals.
In this response, so they're using a life and death situation.
Well, what they found is that when you inoculate... these TRIPs, I guess you can grow in culture, right?
Of course.
And they said when you inhibit these adenylate cyclases in culture, it doesn't really affect the TRIP multiplication very much.
And that's an odd finding, isn't it? Otherwise, it would say, well, what the heck are they using these things for then?
If you have 80 genes for something and you interfere with them, their products, and it has
nothing
no effect
happens in culture.
In the old days, we would say that's a non-essential gene.
Exactly.
Before we knew.
Exactly.
And Harry Ginsberg was wonderful.
Yeah.
He used to be chair of this department. He was a virologist. He hated it when people would knock out a gene in a virus and say, it's non-essential. And he said, it's not non-essential. You just haven't found the function yet.
Right. Keep looking.
And he said, you got to look in animals.
You do.
And a lot of these genes don't function just in cell culture.
Of course they don't.
But when you put them in an animal, there's where you see the effect.
That's right.
Am I preaching too much?
No, you're not. But what you're saying is that animals will always play a role in the development of pathogenesis understanding for pathogens.
So they put these TRIPs into mice.
Right.
They say either with a needle or the bite of a tsetse fly.
Aha.
Why would you do both, Dickson?
Well, to see whether or not there are some changes that might occur inside the fly, which would alter the expression of their changed
It's the natural
gene.
way of it being delivered.
And it's the
after
most
all,
natural
in
way.
nature.
Very few
syringes,
laboratories have colonies of tsetse flies.
Syringes don't walk around injecting us in nature, do they?
I think their mosquitoes are called flying syringes, but that's beside the point. Yeah, you're right. You're absolutely right.
Anyway, they say parasitemia was considerably lower than control.
Isn't that in both the needle and the tsetse fly?
Yeah, for both in the TRIPs that have this dominant negative adenyl cyclase.
So what would be the difference between a needle-delivered infection and a tsetse fly-delivered infection? What's the basic difference there, Vince?
Well, the needle, they're putting an intraperitoneally. And the tsetse fly, of course, is just probably subdermally, right?
Intradermal.
Intradermal?
Yeah, it's right in the top layer of the skin.
Wow. That
So
fly
those are
is really good at it, right?
Those are totally, well, they have to get down into the level of the blood supply.
Uh-huh.
They've got these cutting, they're part of the tabanid fly groups, and
Oh, they have those cutting
they have
things?
cutting plates.
6 legs?
6 legs.
Okay.
6 legs. And cutting plates that are quite painful, actually. To get bitten by a tsetse fly is a painful thing. And so that introduces...
Have you been bitten by a tsetse fly?
I hope not. Because in East Africa, which is the only place I've been, 50% of the tsetse flies are infected. However, there's only one tsetse fly per square mile. One.
Why?
The density is so low.
Do
Why?
they exclude their competitors?
No, because the area is so dry and so grassland, like in the Serengeti, that there's not enough nutrients to keep a larger fly population alive. In West Africa, the Glossina group of tsetse flies breed along rivers where there's a lot of nutrients and a lot of water, so that the densities are much, much higher. And what I'm trying to say is that they tried to say that no matter which way we introduced this trypanosome,
It had the same effect.
we got the same result.
So interfering 50% with the activity of an adenylate cyclase, you get less parasitemia, which would mean parasites in the blood, right?
Yeah, that's right.
And longer host survival.
But they didn't survive eventually, right? They all died. Yeah.
Yeah, they just take longer to die.
However,
they said that when they passaged these trips from mouse to mouse, it's called serial passaging.
Yes.
You take a little blood out, I guess, right?
Yeah.
And then you infect another mouse.
Yeah.
It eventually led to reversion, and they got regular parasitemia and killing. And then when they looked in these trips, they had lost the dominant negative transgene.
Damn.
Right? Because there's no selection to keep it there.
Which one of the 80 did they choose? That would have been a difficult thing for me to actually say, okay, which one do we start with?
I think there is a common catalytic domain.
Okay.
Which is why it probably inhibits all of them, although they just look at total activity.
Right.
Total enzyme activity.
They all might not be on either.
They might not all be on, exactly. Right, but the point is that inhibiting the total activity is enough to get this phenotype.
The point is that I'm really astounded that this worked, because the trypanosome, once it does get into a mammalian host, switches its protein coat to completely cover the surface with this.
Well, this is very early.
But that doesn't matter. It still happens early. But the point is that these catalytic domains are not active for the trypanosome itself. These are inhibitory molecules for the innate immune response. So how does that work?
Right.
How does that work?
There's something in the innate response that is inhibitory, and then perhaps this...
But how does this molecule get to the innate immune response to inhibit it?
Well, we might find this out if we continue.
I'm begging the question because I want the readers to sit on the edge of their chairs.
Well, we have just reached the last page of the paper. So
we have.
There's only so much we're going to find out.
That's true.
So the next thing they did is they want to know what parts of the host immune or innate system is involved, if it is at all. So they started to look in mice lacking various immune components.
It's wonderful to have a series of immune deficient mice to work with, isn't it?
Yeah. And the key one was mice lacking the gene for tumor necrosis factor alpha, which you probably know a bit about, even though you've stopped reading, right?
I haven't stopped reading. I've stopped understanding, but I still read.
You're patient, because most people would have years ago said, Vince, I'm tired of your insults. I'm not doing this anymore.
I don't consider these insults, by the way.
What do you consider them?
Because I can come back to you with equally...
You have good innate immunity, right?
I have a good self-image.
So TNF and TNF alpha null mice. So they had mice that others had made that don't have the gene for TNF alpha.
Right.
In these mice, it looked, even if you put these dominant negative parasites in,
didn't
matter. It grew really well and killed the mice.
Right.
So that suggests that the target of adenyl cyclase is TNF in some way.
That's right. Which is an inducible protein.
It is. It typically you don't have a lot of it, because you'd feel really bad if you did.
That's right.
Because it makes fever and...
Who makes TNF, Vince?
A variety of pharmaceutical companies.
No. What cell in the body is predominantly involved in TNF production?
Tell me.
It could be macrophages.
I think it's phagocytic type cells.
I do too.
And TNF is often one of the early products of the innate response.
Then what happens after that?
Well,
what does TNF do?
Well, it can kill cells.
Directly?
Can kill cells directly by inducing apoptosis. It causes fever, which is a byproduct of infection. What else? What do you want to know, Dickson?
What about the other innate immune response genes?
It's not induced by TNF. Those are all induced separately, if that's what you're going at.
Well, TNF then has a direct effect against trypanosomes?
I don't know. Let's go through the paper.
Yep.
It's induced in many viral infections as well, and ends up killing infected cells. That's one of the ways to get rid of the infection.
If you raise the temperature of an organism, what do?
Tell me.
No, tell me.
Do you reduce parasitemia?
No, so for every 10 degrees of increase in in temperature, you get a doubling of the biochemical reaction.
Okay.
Right? So that's not going to raise it 10
No, it's not going to
degrees.
But it will go from 98.6 to 102 or something like that.
Right. So the rate of synthesis of defense mechanism molecules increases.
So you think that's why the temperature
Fever is a good thing. And the other thing, of course, is that the organism may not be temperature insensitive. It may actually have an optimum growth range.
I'm not sure that that's ever panned out, right?
No, there are some organisms that are inhibited by higher temperatures. And that explains host ranges, by the way. There are some organisms that only grow at high temperatures or low temperatures, right? And some hosts have lower or higher.
Okay. So
that sort of thing.
But for a lot of viruses, it doesn't really pan out.
Okay.
So TNFs are involved.
Right.
Based on that. Okay.
Right. Right. So now it begs the question: what role does TNF play?
Now they did some experiments to look at the amount of TNF that is made.
Right.
So
does it recruit cells to an area? By the way, if it's produced locally, does TNF recruit cells to the area?
I don't think TNF is one of the cell recruiters. There are a lot of other cytokines that recruit cells in. You know, the interleukins and the chemokines and so forth. I don't believe that TNF is one of them. TNF can be made locally, right?
Right,
so
sure.
if you have an infected area, you get TNF production. Then it gets into your circulation and goes everywhere.
Sure.
And that's why you get systemic effects.
Doesn't that induce nitric oxide production?
Nitric oxide synthase is an interferon-induced enzyme, which would be made in response to pathogen invasion.
Yes.
And nitric oxide synthase makes nitric oxide, which is then coupled to proteins, and it inactivates them.
Yeah.
And so it can inactivate viruses or bacteria, but it also destroys your cells, so that's some of the pathology associated with infection.
Right.
All right, let's see. What else should we see here? Look at these experiments here.
You have to sort of come back to the in vivo model and say, if that's the case, what's really going on?
They want to know now what is the effect of these adenylate cyclases. So they know that the product of an adenylate cyclase is cyclic AMP.
Right.
And they say, well, one of the things cyclic AMP is known to do is to activate protein kinase A, which in turn inhibits TNF synthesis.
Right.
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 Before I get to that, I should say that in mice infected with these TRIPs with the dominant negative adenylate cyclase, they measured TNF alpha, intracellular TNF alpha, and in mice infected with the dominant negative TRIPs, it's much higher. It's 2 to 4 times higher than in mice infected with wild-type parasites.
Right.
So that suggests that the wild-type parasites are interfering with the production of TNF alpha because it's bad for them, right?
Well,
and then if you take away the adenylate cyclase, they make more TNF alpha. So somehow you're interfering with the ability of the parasite to interfere with TNF alpha production.
So you can buy a TNF alpha, can't you?
You can buy it.
Yeah, you said lots of companies make it.
Yeah, it would probably help mice fight off an infection if you gave it to them, yeah.
So I have a culture dish here.
Yeah.
And I've got some trypanosomes growing in that culture dish, okay? And the cyclic AMP molecules that they're producing or knocking them out has no effect on the culture ability of the trypanosomes. But what if I threw in some TNF alpha into the culture medium? What effect does that have on the trypanosomes directly?
It's probably been done.
Do they talk about that?
Well,
I said I read the paper, but I didn't encounter that statement.
Let's see. They do mention early on what's important.
Because these are dead organisms inhibiting the innate immune response so that other live organisms can survive. That's where I got this altruistic idea from.
Well, I don't know if it's altruistic or it's just the host responding to the presence of the organism and making things to protect it. So I wouldn't look at it as altruistic.
Yeah, well, no, but I mean from the trypanosomes perspective, the lack of production of TNF alpha by your dead colleagues.
Well, it's by. I don't think it's just by dead colleagues.
You owe them.
It's just by adenylate cyclase.
Yeah, but it can't get to the host unless it's from a dead organism, though, because it's a transmembrane protein. When the organism dies,
You
it's
know,
released.
that's not clear. We don't really know where it's active. These guys are just measuring total cellular adenylate cyclase.
Well, that's my assumption. Then. Maybe
sure
I've made
that's
an
right.
assumption.
No, I don't think that's
I'll
right.
raise it as a question. If they're listening, perhaps they can answer that question for us.
Okay. So TNF is the key. Cyclic AMP is known to be involved with TNF synthesis.
Right.
Okay.
Right.
So they looked at protein kinase A, which,
Okay.
as I said, can regulate TNF synthesis. And they find that increased pKa is only observed in cells infected with control parasites. If you infect with the dominant negative, you don't see increased protein kinase A.
They don't infect cells.
Increased pKa activity is observed only in cells from infected mice.
From infected mice. So that's still, I raised the question of how does that signal get to the host from a trypanosome?
Well, cyclic AMP is a second messenger, so it's running around. It's produced by...
You think it's produced on the surface of the organism?
I don't know where it's produced. I think it's a very good question. We don't know where these things happen.
Because ATP is not usually an extracellular
not all of these,
product.
Not all of these, I understand, not all of these adenylate cyclases are membrane bound. Some are intracellular, so they could be making CAMP there. Then the question is, how does it get out to do its activation of protein kinase A and inhibiting TNF alpha? We don't know.
Yeah.
So to clarify this experiment,
Okay.
you take cells from mice infected with wild type or dominant negative parasites, and you just measure the pKa activity, and they find that increase is only seen in wild type. Wild type infected mice. So somehow this is the mechanism that the adenylate cyclase is affecting levels of pKa, which in turn infect TNF. This is very simple. It could be much more complicated.
Absolutely.
And some of the things you've raised are very valid questions. Where does this happen, and where does the cyclic AMP come from? Where's it going?
But this is just a delaying tactic on the part of the host, because ultimately it's a delaying tactic on the part of the parasite, because ultimately I'd back that up. It's a delaying tactic which allows the host to live just a little bit longer, but ultimately it dies no matter what.
So here's an interesting summary. So our results indicate that about 50% of total T. brucei adenylate cyclase is not essential for growth, but is crucial for regulating processes involved in the control of parasitemia. AC activity, whether triggered upon phagocytosis by the phagosome or by trypanolysis. See?
Yeah.
Okay.
Yeah, so dead trypanosomes.
Reduces the early innate defense against live parasites before the massive release of immunomodulatory factors, such as the VSG protein at the first peak of parasitemia. So apparently, when you get that first peak, you get a lot of release of VSG, and that's really messing with you.
Game's over.
But here the host is trying to intervene early on. And
that's right.
Sometimes it wins, and sometimes it doesn't, depending on your polymorphism, I presume.
So here's a bigger question then, and that is, in East Africa, where this parasite is indigenous.
Endogenous or indigenous?
Indigenous, depending on your British or not upbringing. All of the animals of East Africa have this infection. All of them. Mm-hmm. None of them die from it. None. Zero. Except introduced cattle from Europe. So there's some other innate immune response that protects these animals from dying from an infection that they've lived with throughout their evolutionary history. But that's where we come from too, Vince. We come from East Africa, right? Come from the Olduvai Gorge region of northern Kenya, southern Sudan, Ethiopia border. And it's amazing that we didn't evolve similar mechanisms for our protection, right?
As the animals there, you mean?
Yeah, exactly.
I don't know. It's a good question. Well, I mean, many people left, and so there's no more pressure.
Many people left. There weren't many people, but the
there
ones that were there left.
There are people who live there that haven't been able, and maybe it's not compatible with human survival. As opposed to the next story that we'll talk about, there is some...
So, people don't live in Africa where trypanosomes are dominant. They don't live there.
No?
They don't. They do not.
So when you get infected,
it excludes people from large zones of Africa.
But why do people get infected then?
No, because I said there's one fly per square mile, And occasionally a person does get infected in East Africa. In West Africa,
maybe
common infections.
Maybe
in
it's
West
not
Africa.
enough pressure.
In West Africa, common infections, but that's not where we're from. Yeah.
All right. Is that
Okay.
good? Are we okay with that?
That was a great paper because it leaves a lot of unanswered questions, but very interesting
Pretty neat.
questions to answer in the future. And we look forward to more results from that group and others too.
Keep your eye out.
Exactly.
But you don't just look on the cover of Science, I mean. Now, Cell is our next paper.
Okay.
Actually, it's Cell, Host, and Microbe.
Ah.
You know, these journals are
They're starting to
replicated.
specialize now.
They just make more and more money, so they make more and more journals.
See that? Is that what it's all about?
Well,
Vince?
you know, many years ago, you had Science and Nature,
True.
and then Cell came in
Yep.
the 70s.
Yep.
And they found they could make a lot of money. And in fact, they only could publish about 1% of the manuscripts that were submitted. So they started multiplying. And there are many, many Cell journals now, and Cell, Host, and Microbe is one of them. And I find it discouraging because it's a huge money-making practice, and scientists just want to publish their data.
True.
And the idea that it's a profit-making enterprise, I think, interferes with it. But we've talked about this on TWiP. Anyway.
So we have infection and immunity. We have a lot of other journal choices to submit articles to that have similar themes. And you might have about 8 or 10 journals that trap that research group. But there is a hierarchy.
Yeah, there's always a hierarchy. There's some
Cell,
that are considered high.
it's
Cell
the
is
highest.
considered a very good with Cell, Nature, and Science, and many others are considered good. And if you publish in them, it increases your chances of promotion, tenure, et cetera, et cetera, et cetera. So these journals are determining our career decisions. It's really
interesting.
It's really unfortunate.
We should have a discussion about that someday.
Yeah, we often touch on it on TWiP, and I think a good discussion would be nice. The future of publishing. I mean,
right.
There has to be some other kind of
Well, there's
model.
a bunch of open journals, like the PLOS journals.
Yeah,
right?
so there are many open journals, but those are also still competitive.
They are.
That's fine, and they have other ways of making money.
Yep.
But I think you just need a place to publish your data so that people can see it and eventually evaluate it, independent of where it's published. I don't think where it is published should make any difference.
But it's how it's
quality
published.
of the
yeah,
work.
That's true.
So why should an editor at a journal have a say in your tenure decision? It makes no sense whatsoever. I think the work should stand on its own. The review process is flawed.
And it's also based on how many times you're quoted in the literature and things like this.
Impact factor of these journals is based on citations.
I know.
And all that. Anyway, this is a Cell, Host, and Microbe article. It's called Translation... Sorry, wrong word. Translocation of sickle cell erythrocyte microRNAs into Plasmodium falciparum inhibits parasite translation and contributes to malaria resistance.
Right.
A lot of information in that title, Dickson.
I'll say.
First of all, what is a sickle cell erythrocyte?
Well, I mean,
I know you know all about sickle, right?
I don't know all about it, but I know a lot about it. At least I did.
Tell us what you do know.
Well, there are hemoglobins come in many varieties and flavors and kinds, okay? And so hemoglobin...
Which is the protein in your red blood cells.
That's right. That's right.
Which you need to carry oxygen around.
That's right.
Okay.
So and to deliver CO2 to
How many chains in hemoglobin?
I think there are...
More than one?
Yeah, there's
So you
more
could
than
do a
one.
dominant negative, right?
You could do a dominant negative on this
Just
one. That's right.
Making sure we reinforce what we've learned here.
The porphyrin ring is the center part of the hemoglobin molecule. And instead of cobalt in chlorophyll, it has iron. Okay? But ironically or interestingly, these two molecules, which rule the world for aerobic metabolism and photosynthesis, take advantage of the same basic porphyrin ring structure. Okay. So I'm astounded by that. It's the protein globin portion that varies.
Right.
And so hemoglobin F, which is fetal hemoglobin, is the one we produce first. So hemoglobin A is adult hemoglobin. And hemoglobin, normal hemoglobin, in all of its infinite varieties, is the food that the parasite eats. Okay, so you can watch it being eaten by looking at the degradation of hemoglobin in the parasitophorus vacuole, and it's totally dependent upon cathepsins. And remember, when we discussed malaria, we discussed how it can't digest the porphyrin. Right. So it has to find some place to throw it away.
Right.
So it has this stacking enzyme that it uses to get rid of it. And eventually it just leaves it behind in the host cell as it divides and gets out, right?
Okay.
So that's known as malaria pigment. So just to bring the audience up to date as to how the normal parasite handles normal hemoglobin.
Talked about that.
We did.
And
so
we had
now
4 episodes on malaria.
We did, and we had some people on. If we had David Fiddick here, and we had some other people and Tom Willems. Did we have Tom Willems?
No, Tom Willems.
We meant to have Tom Willems then.
Yeah.
The point is that there are things known as hemoglobinopathies.
Mm-hmm.
Okay, it's a pathology associated with abnormal hemoglobins. Mm-hmm. So you have diseases associated with them, like sickle cell anemia, thalassemia major, thalassemia minor.
These are mutations in the hemoglobin genes, right?
Yes, not the porphyrin part,
right.
The globin part.
Because we know what these mutations are.
That's correct. So I said that there were several chains, but there's only one.
There's one?
There's just one. There's the globin. And in fact, when the organism clips off the chain, it leaves a little peptide behind. That's part of the porphyrin ring.
Why did you say it was multiple chains?
Because I made a mistake.
And why did I agree with you?
I don't know.
And I'm glad you corrected yourself.
Well, I started to think about how this...
Well, there are alpha and beta subunits.
Oh, okay.
Right?
Okay. So the point is that when you start making amino acid substitutions...
We're going to both talk at the same time. No. Wait, how many chains are there, Dickson?
You tell me now. You've got the internet here. That's not fair. You're stretching my biological knowledge back to the abyss that everything has found itself into.
It's structure.
Well, the porphyrin ring, and then it's globin. So the globin part has how many parts to it?
4 globular protein subunits.
There you go. One off of each part of
there are 4 subunits, so it can be attacked by dominant negative. But that's not the point of this paper at all. And I'm sorry to divert you.
No, that's okay. You're just
sickle cell.
You're showing me how shallow my knowledge actually is in this whole subject.
What's sickle cell?
So sickle cell, hemoglobin S, all right?
S stands for sickle. Okay.
Is inherited from a parent.
Okay, it's a mutation in the hemoglobin gene.
Yeah, so
right?
Male and female people can have hemoglobin S or hemoglobin normal and hemoglobin S mixtures.
You can have 2 copies of the mutant gene.
Or one.
Or one. If you have 2, is it lethal?
It's horrible. It's a horrible disease, and the reason why it's horrible is because it can't carry oxygen in the normal sense. Once it reaches a lower than oxygen level that's normal in the blood, the hemoglobin starts to crystallize. And when that happens, the cell deforms to form this sickle-shaped cell. And that's where the name comes from. So when you reduce the oxygen content of a hemoglobin S double mutant carrier, their red cells start to deform, and that creates problems in the capillary beds because a deformed red cell that's in the shape of a sickle doesn't pass through the capillaries very smoothly. All right?
But if you have one mutant gene.
You have one mutant gene, the red cells do not deform, but it's still half as good as it would have been if it had just normal hemoglobin.
And these sickle cells are resistant to malaria?
Yeah, the interesting part of this whole thing started a long time ago when it was noted that people with either the sickle cell trait or sickle cell anemia,
both 2 mutant genes, right?
are more resistant to the lethal effects of Plasmodium falciparum than people without either one of these 2 hemoglobins to protect them from that. And so that began the quest to find out, how does that work?
I think we talked about this, right?
We did, because in William Traeger's laboratory at Rockefeller, who was a world-leading expert on the molecular biology of malaria in his time, he discovered that when the red cell deforms with a parasite inside, the crystal of hemoglobin S goes right through the parasite. It actually sticks right through the parasite and kills it. So it's like a sword, right? But he was hard-pressed to show the killing effects of normal hemoglobin S red cells, which don't deform.
Right.
So he didn't have a good answer for why that happens, although he could relate it to intracellular ATP levels in some way.
Well, according to this paper,
yes,
there are multiple mechanisms by which the sickle gene confers protection against malaria.
Okay.
But it still doesn't explain it totally. And some of these
doesn't.
Mechanisms, do you want to go through a few of them?
No, you can do that.
Well, you could just say yes or no.
No. Why don't you refresh our memories in terms
parasitized mutant polymorphic
Yeah.
erythrocytes have been found to be subject to enhanced phagocytosis.
Okay, because they deform and therefore they're more, I guess, visible to the immune system.
Progressive dehydration of erythrocytes and increased cellular density are associated with decreased invasion.
Okay.
So structural things.
All right.
The hemoglobin allele shows it provides resistance by altering the cell surface properties of infected erythrocytes, resulting in abnormal display of a protein leading to decreased cell adhesion and reduced hemagglutination.
Because Plasmodium falciparum induces a histidine-rich protein
PFEMP1,
under the surface.
Yeah,
and it actually is the basis for the adhesion.
Oh, I see. Okay, so it can't get in as well.
Right.
And finally, P. falciparum grows poorly within HBSS erythrocytes.
Right. It's probably suffering from a nutritional deficiency, but
they don't know the basis for that. And so, in fact, that is what they're trying to find out.
Exactly.
What are the intrinsic factors that are contributing to resistance? Okay. So what they thought in this paper is let's look at microRNAs. Because microRNAs are known to differ in wild-type versus sickle erythrocytes. But let's explain what a microRNA is.
And I'm sure you're going to do that, Vince.
A microRNA is a hot RNA. It's a really small RNA produced by...
When you say hot, what do you mean?
A lot of research. Well, it got a couple of Nobel Prizes
I see what you
a number of years ago.
It's a target for research.
It's a brand new discovery in my career. It was discovered as I was working here.
And they're not involved in translation?
They can be.
They can be.
MicroRNAs are produced in mammals. They're also produced in other organisms. They are short RNAs, 21 to 25 nucleotides long.
So that's divided by 3, it's only about 7 peptides.
No, it doesn't code for anything. It's
Nothing.
A non-coding RNA.
Really?
Yes.
The triplets aren't there in those microRNAs?
You could translate it on the paper, but they are not translated into protein.
Okay.
So this is important. They are non-coding RNAs.
Okay.
Really small. We always threw them away because we would run gels and whatever.
They're not pieces of other larger RNAs.
We would see these things at the bottom of an RNA gel. Who cares? Crap.
Exactly, crap.
But it turns out that they are very important microRNAs. So they are produced as a primary long transcript, okay? And then they are chopped up by enzymes. RNases. They're called Drosha and Dicer. Remember, Drosha and Dicer.
Sounds like these are the investigators' names.
Nice. Dicer because they chops it up. Okay. So it's chopped up from a long RNA precursor into small ones. And then these small RNAs have targets on other RNAs. So wherever they're complementary, they will bind to another RNA.
And then inhibit them from doing their job?
Well, they can either cause the RNA to be degraded.
Okay.
Or
ubiquitin comes in and...
no, not ubiquitin. That's a protein degradation thing. There's a whole system of RNA degradation.
A ubiquitin-like system.
Well, it's not ubiquitin-like at all. It involves nucleases of various sorts. It's very interesting. RNA stability is totally fascinating, but we can't talk about it now.
Well, we are talking about
it. So these microRNAs bind typically to messenger RNA, and then depending on how good the homology is, they can either cause degradation of the mRNA, and of course, if you degrade an mRNA, it's not going to make protein. So you're basically silencing it. So this is why it's called RNA silencing. Or it can inhibit translation of the mRNA without degrading it.
By somehow interfering with the ribosomal translation.
Yeah, so there's a lot of work going on to figure out how that works. It's a little bit controversial.
Why would we need that system, Vince? Why would nature select the system?
Well, so we make a lot of microRNAs ourselves, humans, other mammals too.
Yep.
They control our genes. They're a way of regulating our genes.
That's incredible.
And they're also involved in immune responses. It's been shown they're highly involved in repelling invaders.
Wow.
Okay, so they're multifunctional.
So how many genes do we have in our genome that encode for
microRNAs?
Yes.
You know, this morning I woke up and I said, you know, Dickson's going to ask me how many human microRNA genes there are.
Right, because these will be big genes that encode multiplicities of microRNAs, right? So one gene could encode 5
You could
different...
have 1 or 5, but there are 1,000 microRNAs in the human genome.
1,000?
1,000.
MicroRNAs. So how many big pieces does that...
those are embedded in longer transcripts.
So genes would be maybe 100?
It could be. I don't know how many genes that... but it targets 60% of mammalian genes.
Wow.
So those 1,000 microRNAs target 60% of our genes.
That's wild.
To regulate them.
That's wild.
And they're also involved in limiting pathogens as well.
So you said somebody got a Nobel Prize for working on this?
Yeah, I did. Do you want to know who that was?
Of course we do. Listeners have an inquisitive mind on this subject.
Right. Their names are not at the tip of my tongue.
No.
But let me look it up here.
Okay.
I know who didn't get one who wanted to.
Well, there's a big list of those people out there, I'm sure, for other subjects as well.
Okay, here we go.
Yes.
2006. Nobel Prize in Physiology or Medicine was awarded to Andrew Fire and Craig Mello for the discovery of RNA interference.
Okay, that's RNAi.
Yeah. So RNA interference is the broad term for interference at the level of RNA. It turns out there are many kinds of small RNAs in cells that do this, not just microRNAs, but there are many others.
All right.
Now, let me just tell you something.
Please.
Andy Fire was a graduate student when I was a postdoc at MIT. I used to see him walking up and down the hall, this wacky guy. He was a nice kid, and he was really smart. I never thought he'd get a Nobel Prize.
Neither did he.
You never know who's going to get a Nobel Prize.
Sometimes you do know, though, I guess.
But
that's amazing.
Really interesting. So they worked in C. elegans, and they discovered it there.
Nice.
Now, there's another player here, Victor Ambros, who was an early discoverer of a regulatory RNA in C. elegans, and he probably should also be acknowledged to some extent, but he hasn't been.
Oh, too bad.
Anyway.
Right. So now...
So worms make microRNA, small RNAs, plants do.
Apparently everything does.
And plants, they're heavily involved in antiviral resistance.
Sure.
Insects, it's a huge component of the insect immune response, these small RNAs. Yeah, because insects don't have adaptive responses.
I wouldn't go that far, Vince.
They don't have antibodies.
They don't have antibodies, but they do have cells that actually increase in numbers as the pathogen.
Yeah, but they may make microRNAs.
They may.
Anyway, you know what? Whenever you make a broad statement in biology,
you can find an exception.
There's always going to be an exception.
Of course.
In general, we think insects have a really well-developed microRNA or small RNA inhibitory system because they don't have anything beyond that. Right. But they have toll-like receptors, but they seem to be involved in pattern development.
Okay.
And also innate recognition. Right. So in this paper, they wanted to see if microRNAs played a role in the resistance of sickle erythrocytes to Plasmodium falciparum.
Right.
And why would you do that? Because people had found that there are differences in the populations of microRNAs. Sure. If you compare a normal erythrocyte with a sickle erythrocyte.
What's the beauty of this system?
Which one? The Plasmodium?
Yeah, you can do it in vitro.
Is that right?
Yeah, you don't need a host.
You take blood cells?
Yeah.
And you put parasites and they grow in them?
Yeah, and so guess where that was perfected?
Traeger.
That's right, and Jensen.
With the candle jars, right?
That
in fact,
correct.
They quote one of his papers, 1999. It must have been one of the last papers.
It wasn't the Traeger-Jensen paper, though.
No, but he was still alive in 99, right?
Of course. That's right.
But he's no longer, right?
No, unfortunately.
Well, everybody dies.
I know, but he was a nice man, and I enjoyed talking with him a lot on the phone.
So are you saying that people who are not nice deserve to die?
No, but they're just not nice to talk with, that's all.
Right, so
this whole system takes advantage of an early finding. That's all I'm saying.
What they did is ask when a malaria parasite infects an erythrocyte, and we know there are microRNAs in the erythrocyte. So the thesis here is that a human microRNA is somehow going to inhibit the malaria parasite.
Right.
So they said, are they getting into the parasite? Okay.
Correct.
So they infect erythrocytes, and then they purify out the malaria parasites that have grown in there. And they ask, are there any human microRNAs in them?
That's right.
So it's a weird thing. Are they taking up microRNAs? And it turns out they do.
Do they take them up from the normal cells too?
What normal cells?
Normal erythrocytes. You have to have controls.
Oh, yeah, they do. In fact...
so that means the pattern of microRNAs is different from sickle cell cells to normal hemoglobin-containing cells.
Yeah, they do actually take them up. So they determine whether erythrocytic microRNAs are present in the parasites.
Right.
Okay?
Right.
And they found about 100 microRNAs within the parasites. A couple of them were enriched. Okay?
Right.
There are 3 that were particularly enriched. And that is because they're enriched in the erythrocytes. So whatever is enriched in the erythrocyte gets translocated more frequently into the parasite, which makes sense, right?
Well, not necessarily. If there was a transport molecule, then it could have favored one over the other, but it looks like it's passive.
I don't know how it would do that. It's just an RNA. They're all short RNAs. You know that?
There's only one way for anything to get inside the malaria parasite, and that's through the food vacuole, period. So it creates this large extracellular vacuole that it sucks up the hemoglobin into. So part of that soup that it sucks up must have these interfering RNAs in it. And how does it get across the membranes? You're suggesting passive because it's just dependent on the amount of each present as to what gets in.
That's what they suggest, that whatever is enriched in the erythrocyte, it gets enriched in the parasite. So it's probably some passive mechanism, right?
Yeah.
So this is normal erythrocytes. So they have their complement of microRNAs, and they get translocated.
But they don't have any effect on the parasites.
Apparently not. Well, that's the story now. The point is going to see. You will see that there are some enriched in the sickle that are particularly inhibitory to the parasite. How about that?
That's an amazing selective pressure.
So they showed
evolution.
Previously that there are 2 specific microRNAs. So they're called mirs, microRNA, and then they have a number after them.
Okay.
So they're 1 to 1,000 in humans. So number 451 and 223 were elevated in HBSS erythrocytes. Amazing. Those are with 2 copies of the sickle, right? HBSS.
Yes. That's
so
right.
they found that the HBAS also have elevated forms of these 2 microRNAs. Okay?
Amazing.
Which is now making a story. And so
it's a very interesting story.
They figured they took these 2 microRNAs and they made them and transfected them into plasmodium cells
and inhibited their development.
Yeah, basically
remarkable.
It inhibits their replication.
So does this suggest, Vince, that we could actually use this as a therapy?
Yeah, it does, absolutely, because a lot of people are exploring microRNAs therapeutically for many things, diseases, infections. So they've identified 2 microRNAs that inhibit parasite growth, and yet you could think about delivering these to people with malaria to inhibit growth for sure, maybe in conjunction with something else, right? Sure.
No harm, no foul, right?
Absolutely. So they say that these microRNAs end up in the parasitophorus vacuolar membrane.
That's right.
Is that what you were saying? This is how they take up stuff?
No, the parasitophorus, actually, when you look at a red cell, how could you call that a phagocytosis event? When the parasite enters the red cell, it doesn't actually go inside the red cell. You know that. It actually...
No, it stays in a vacuole.
It deforms the membrane, and the red cell actually doesn't lose the size of the original red cell. So it must have spare membrane proteins to accommodate the involution of its membrane and accommodate the parasite. Okay. So that when you extract the parasite from the red cell, it's got an extra membrane on the outside, which is the membrane that it pushed in from the erythrocyte when it first invaded it. So you'd call this vacuole, it is the parasitophorus vacuole, but it's not the phagolysosome of a normal invading cell. So it's a parasitophorus vacuole.
It just says it's in the same area as the parasite, right?
Right, but when the parasite starts to feed, it breaches this membrane, all right, and it creates a hole which feeds into its own food vacuole.
Okay.
So there's this little pore that it develops. Maybe there's a pore-forming protein that the parasite secretes to make this happen, right at that little spot. And at that point, the microRNAs can gain entrance into the parasite.
Okay. So they put these microRNAs into parasites. They have 3, 451, 223, and another one,
Right.
It's called LET7I. They all can inhibit malaria reproduction.
It's amazing.
And they have some controlled microRNAs that have no effect.
And the equivalent in a normal red cell is different in composition for the RNAs?
Yes, these are not enriched. That's it. They get enriched in the sickle, and they're inhibitory.
That's incredible.
It's really weird. I don't know if it's an accident or if it was something that was selected for.
It's a selection. Got to be a selection, Vince.
You think?
Yeah.
I don't know. Maybe
because where does the hemoglobin...
Why would a sickle have these microRNAs in abundance? Maybe it has something to do with...
Let's talk about who gets sickle cell anemia. Who gets it?
Well, let's finish the data first.
Okay,
Can you hold it?
Sure.
So these particular microRNAs can inhibit plasmodium growth. They're enriched in sickle cells. Okay? That's the observation. So this partly explains why sickle cells are resistant to malaria, because they have these enriched microRNAs that get into the parasite and they inhibit. So how do they inhibit?
Yeah, right.
All right. So normally a microRNA...
This is fascinating part too.
Hybridizes to messenger RNA.
Hybridizes.
So they started looking. So what you do is you look for targets of microRNA. It's really hard to do because these are short, and the bioinformatics of finding microRNA targets is a nightmare because you find a lot of targets and you have to validate them, and that's hard. You have to do experiments.
Can you make an RNA gel?
No, you can't do that. Absolutely not.
Okay, because otherwise it'll degrade, right?
Well, you could do a northern blot, but it doesn't tell you which gene is the target because all you see is a band on a gel. If you can hybridize with a microRNA probe, but you'll see a band, but you don't know what gene it is. That's the point.
You couldn't cut that out and sequence it?
No, people don't do that. I think this is very difficult. What they do is they take the genome, which is sequenced, and they look for the microRNA complementarity. Okay? And what they basically found here is that there are no clear targets, but the sequence seems to be... they found parts of these sequences in some of the RNAs when they started sequencing the parasite RNAs. And,
right. That didn't make sense then, right?
It didn't make any sense. It was only part of it. And basically what they end up finding is that these microRNAs, these human microRNAs, are incorporated into the parasite messenger RNAs at the 5' end.
In other words, it's a hybrid RNA molecule, part parasite, part host.
Yeah, so these microRNAs get into the parasite.
They integrate it into the messenger?
They get stuck onto the 5' end of the messenger.
And then they form a cap, or they don't form a cap?
They don't get capped, so they're not translated. But, Dickson, what's really weird is that they're only on specific messenger RNAs, not every messenger RNA.
And what did that messenger encode for in the parasite?
Cyclic AMP-dependent protein PKAR.
Have we not made a connection?
We just talked about it, and the other one is regulating TNF. So that's the connection you want to make between the two. Now, I want to tell you why they did this. It turns out that parasites don't have dicer. They don't have the enzymes that would allow microRNAs to inhibit their activity. Okay, but we know from the work that's been done here already that these microRNAs can inhibit plasmodium replication. So there has to be another mechanism. So this is what it seems to be. The
TNF.
microRNAs are stuck onto the 5' end of certain parasite mRNAs.
Yeah.
It prevents their being capped, and the cap is needed for translation, so they don't get translated well.
And then they can't induce TNF alpha.
Well, that's trypanosomes where the TNF alpha was involved.
Well, cyclic AMP is it over here?
Yeah, but there are a lot of targets of cyclic AMP.
Yeah, but let me just tell you one of them for Plasmodium falciparum. When the red cells attach to the precapillaries in the brain, they induce TNF synthesis on the other side of the host. In the cytoplasm of the host cell next to the endothelial cell, which then induces nitric oxide production, puts the patient to sleep.
Yeah.
So you go into a coma.
Yeah, I remember that from another TWiP.
Right. So if you can't do that, Maybe this has some connection. Maybe. I'm just speculating.
So basically
here.
they show now that if you have a lot of the specific microRNA in the erythrocyte, it gets into the parasite and it modifies this mRNA. Right. You take away the microRNA, you don't get mRNA modification.
Right.
You take away, they knock down some of these microRNAs and they can show that you don't get these chimeric mRNAs.
So there are other blood dyscrasias. Do they discuss them as well?
There are other what?
Blood dyscrasias, they're called.
What's a blood dyscrasia?
It's a pathology term for hemoglobinopathies. So
I
like
didn't talk about it at all.
So like thalassemia, for instance. There's some selective pressure there to encourage survivability with Plasmodium falciparum throughout the Southern European areas like Italy and Greece and
no, they didn't talk about it at all.
Not at all.
All right, so PKAR, protein kinase A R. All right, and again the name. Let's go through the name again so that everyone remembers.
Right.
All right. PKA, protein kinase A.
What's the R stand for?
Hang on. Well, there are different isoforms that have different activities.
Okay.
Man, this is a long paper. Cyclic AMP-dependent protein kinase A. R is the regulatory subunit.
Okay.
Okay, so this dimerizes, and there's a catalytic subunit and a regulatory subunit. So this microRNA is down-regulating the amount of the regulatory subunit.
Right.
Okay.
Which
and that has an
enables Tnf alpha to perhaps
No. Stop with the Tnf alpha.
Go away from
TNF
that.
Alpha.
All right.
I'm going to tell you what happens here. So you reduce the amount of PKAR, and that gives you increased PKAC, catalytic, which is the active form of the enzyme. So basically, the microRNA is activating PKA.
Which then does what?
Because it's removing the regulatory subunit, so it's activating PKA.
That's
okay,
nice.
So now that's the question, Dickson.
Yes.
Right? Modulation of PKAC plays a major role in the parasite's life cycle. Both PKAC and the regulatory subunits have been identified in the parasite, and regulation of R is crucial for parasite survival. So they speculated that the suppression of the regulatory subunit should result in increased catalytic activity subunit, and this has been implicated in the induction of gametogenesis.
Gametogenesis.
Traeger and Gill, 1989.
Yeah.
He worked on that?
Yeah.
So they looked at the amount of sexual stage parasites in erythrocytes with MOC, MIR181 or MIR451. MIR181 is not effective. MIR451 targets PKAR, where we observed a 2.5-fold increase in parasites entering sexual stages if you have normal amounts of MIRs, but if you put the MIR451 in, you get a decrease in the amount of parasites reaching these sexual stages. And that's because you're activating PKAC. So somehow this is involved in gametogenesis.
Well, okay, then I would raise the question then. That has nothing to do with the pathology in that particular individual. Nothing.
Why?
If you made no gametocytes whatsoever, you'd still die from it. Because the gametocytes don't hurt you. That's the only way to pass on the infection to another individual.
Well, you also you also get an effect on
a diminished growth of the parasites.
Too.
So this may not be
maybe two things, right? Okay, so you get this. The microRNA is affecting the regulatory activity of PKA, which allows increased PKA activity itself. PKAC, and that is inhibiting parasite growth and gametogenesis in particular. So the gametogenesis may not be the only effect of it. But so we don't know exactly how this works, but they conclude that translational inhibition of PKAR has profound effects on parasite biology and survival.
Right. It would be more than fascinating. It would be a major breakthrough in chemotherapeutic approaches if you could identify a microRNA which prevented gametogenesis.
Mm-hmm. You
could stop the whole infection.
Could make a synthetic one if you knew what genes are really important.
That means that people who caught malaria could never pass it on to a mosquito.
Yeah. The problem is, Dickson, that microRNA is not going to work in P. falciparum because it doesn't have the enzymes to chop up the target.
You'd give it into
But I don't know if all microRNAs would recombine with the mRNAs as this one have. It's really interesting.
Yeah, would you use this one? I would pick this one as a potential therapeutic agent to begin with, and just make it.
Yeah, this one would be okay.
I mean, I wonder what other...
P. falciparum lacks orthologs of dicer and argonaut. So dicer and argonaut proteins are involved in the degradation of the target mediated by microRNAs. So you can't just put any microRNA into P.
No, I didn't mean any one. I meant the one that they identified as the dominant one that inhibited the development of gametocytes.
So what needs to be done is to figure out what property of that microRNA allows it to become fused to the messenger RNA. Because this is a totally novel, brand new mechanism of microRNA action. You know?
Is that an enzyme that does that?
They don't know. They think it's recombined, maybe recombination enzymes, maybe it's splicing and you don't need an enzyme. You know, splicing doesn't require an enzyme. So this is totally new, but it also gives you a possible therapeutic target.
Sure.
Which is pretty cool.
Absolutely.
But no TNF here, man.
No.
Okay,
forget it. Okay. That's out
so I think the most interesting thing is that this microRNA is working in a new fashion, never described before. And it just goes to show you got to keep on looking in all systems and you're going to discover amazing stuff.
Here.
I think we've only discovered about 1% of everything that's out there in terms of biology.
I would agree.
What do you think?
Or maybe even less. And we've got genomes already, but that doesn't help you understand how they work.
It's pretty cool.
Very.
It's a good paper.
Very.
I hope our listeners got all that. It's a bit complicated.
I mean, P. falciparum's genome has been sequenced also, you know.
I know. And that's why they looked in it. They didn't find a target for this.
Right.
And I'm not surprised because, as I said, the usual way of...
It's hybridization, right?
Is to hybridize to a target.
Not by linking
But only when they started sequencing the RNA, not the genome, or looking at the genome sequence. So you have to actually sequence the RNA directly, then you find these things.
Remarkable.
And if you sequence the RNA from a plasmodium that's been treated with the microRNA 451 or whatever the number is, then you see the whole microRNA stuck on the end of the RNA.
Right.
And if it's an untreated parasite, you only see a little bit. So
very cool.
Should you do a couple of emails?
Sure.
All right. So we have a couple from Robin. Robin has written a lot of emails to us. Big fan.
This is great.
And so I'm saying to Robin, we'll get to them eventually. I'm going to do two here.
Right.
And the first one, he's providing us a definition of trematode.
Good.
Trematodes are commonly referred to as flukes.
Yes.
This term can be traced back to the Saxon name for flounder and refers to the flattened rhomboidal shape of the worms.
Nice.
Also, Robin writes, with respect to men stealing meat from lions,
yes,
check out this video on YouTube, which is called, it's a BBC video.
Yeah.
It's called Men Stealing Meat from Lions. Do you know why he sent this to us? Because you were speculating. We had a letter last time from a listener. Is this a way of getting parasites? Is this the way we originally got a parasite from a lion? You don't remember?
Well,
no, I'm sorry. From some kind of meat, then we would be competing with the lions. You don't remember?
Yes.
You look like you don't remember at all. Do you want me to look up the original email?
Are you worried about cysticercosis or trichinosis?
I'm not worried about anything. I'm worried about you, Dickson.
You shouldn't worry about me, Vince.
Last time we had a letter from a listener, and I'm going to pull up the letter right now. And he asked...
It's a funny letter, huh?
Let's see if I can find it here. Be all the way at the bottom. And you thought it was a good idea, and now you've forgotten.
Well, I can't remember everything, Vince.
Yeah, you can.
No, I can't.
Here we go. Oh, that's the fish tapeworm.
Mm-hmm.
Ticks.
See? Vince forgot to.
Worm in the eye. Oh, here we go. Peter, watching Origin of Us on a BBC documentary on human evolution. It appears that the lion tapeworm and human tapeworm are very closely related.
Oh, okay.
Do you remember?
Okay.
It's assumed that around this time humans began to steal or eat the same prey as lions.
Right.
And eventually the worm evolves from humans as the intermediate.
Stealing meat from lions is tricky business.
Well, that's why Robin sent us the video, because it's all about men stealing.
Shows the Maasai actually doing it.
Yeah, you can do it. It's not easy.
I'm not saying it can't be done. They still do it, in fact.
All right. The next email is from Leesbath, who writes, Dear Vincent and Dickson, let me start thanking you for encouraging me to carry a healthier life. Since taking the bus to the lab each day meant a 20-minute ride, and your podcasts are usually longer, I decided to start waking up earlier and walk every day instead. That way I can hear the whole episode. I feel somehow infected by a parasite podcast that modifies my behavior in order to be listened, reaching its objective when I arrive at the lab. What?
Just turn around a minute, Vince. You're going to see a recreation of a World War II scene. Out the window is a Mustang P-51 following a B-29 bomber. Don't know why.
Wow, what's going on today?
I don't know. Maybe it's a reenactment of World War II.
Why is he following it?
That's a good question.
Not being fueled,
no, because they were escorts.
I see. Cool.
And bombers could fly longer than most of the attack fighter planes, except when they invented the P-51. Had a pod on the bottom to carry more fuel. So they could accompany them all the way to Germany and all the way back again. Okay. The Luftwaffe didn't have a chance.
That's cool. Yesterday we saw a Navy blimp, remember?
I do. So
Something's going on.
Something is going on.
I feel somehow infected by a parasite podcast that modifies my behavior in order to be listened to, reaching its objective when I arrive at the lab, commenting on the last episode I heard. That's cool. I like that idea.
Yeah.
After your discussion on the origins of the term Loa Loa, I did some internet research and came up with an article published in 1991 by Ruby and Hall. And she quotes from it, and that's in fact the article we read last week. I think another listener sent that in.
Right. Good.
So thank you very much for sending that in. Looking forward to your next infectious TWiP. Todd also said, found the following reference for the origin of Loa.
All right.
And he sent a Google Books search, which has a bunch of references to Loa. So people got very interested in our query, what does Loa mean?
Indeed.
So there you go, 3 people. Todd is a lab tech at Georgia Perimeter College.
Oh. Must be on the outskirts of Georgia.
That's right. You are so funny. Peter writes, just to get you started, grin. And this is an article. This is very funny. On Scientia Rules, the origin of the human tapeworm. Look to lions, not lambs, for the origin of the human tapeworm. So this is a similar story as we've been talking about. Yep. Tapeworms. Oh, another plane?
Yep. This is a B-17.
Wow. You know all these planes?
I do.
Cool. Now, these are all propeller planes, right?
Yeah.
Neat. There must be some kind of a show.
I believe there's got to be an air show someplace, but it's great to see these old airplanes still flying.
Tapeworms that plague humanity originated in carnivores such as lions and hyenas and jumped to humans after we began eating their prey animals on the African savanna. So this is an article that discusses this, and it's actually based on a Proceedings in Biology and Science article from 2001.
Are they talking about Taenia saginata or Taenia saginata and Taenia solium?
They're talking about Taenia saginata, Taenia asiaticus, and Taenia solium.
Okay. Because the warthog would be the intermediate host for the pork tape.
Right. Well, as they say here, science has traditionally adopted an anthropophilic view in which humans acquired T. saginata and asiatica from ancestors of cows,
right.
T. solium from ancestors of pigs,
right.
Following the domestication of these species. But Hoberg's work turns this on its head. One of the closest relatives to T. saginata and T. asiatica is T. simbe.
Right.
As you might guess from the name, this is a tapeworm whose definitive host is the lion.
Yeah.
So there you go. So this further emphasizes what we've been talking about. It's a cool article. I'll put a link to that.
Yeah, nice.
You should read it.
Nice. It says a lot about human behavior, too. When you're hungry enough, you'll try to steal prey from a lion.
Alice,
and,
yes.
By the way, I must also say that the video showing Maasai chasing lions off of meat runs against the commonly held belief that Maasai only drink milk, urine, and blood, and they don't eat meat. They actually do eat meat.
Mm-hmm.
Yep. I've actually been in that area once and saw these people, which are remarkable people, by the way, the Maasai, living in harmony, apparently, with all these prey animals that inhabit the Serengeti. To see someone walking without protection except for a spear among the the wilds of the Serengeti is quite sobering. Imagine what it would be like if you did that.
I don't need to go there.
I would just have to run as fast as I could, but I wouldn't escape the run of a cheetah or a lion. I don't think cheetahs eat people, but I think lions regularly do.
Our next letter is from Alice, who says, did you see this about Toxoplasma gondii? It's an article saying female owners of cats are more prone to suicide.
Really?
Study found that women infected with T. gondii were 1.5 times more likely to attempt suicide than those who are not infected. The study involved more than 45,000 women in Denmark, published in the Archives of General Psychiatry.
So much for pet therapy.
Well, today I saw an article in Science magazine.
Yes.
It's called Cats Don't Cause Cancer. It says, last year, cat owners got a scare when a team of French researchers reported a possible link between felines and brain cancer as a consequence of T. gondii. Another team of researchers believes it has settled the issue. The scientists examined 600,000 British women aged 50 and older and tracked how many developed brain tumors over 3 years. 18% of the women, over 100,000, owned at least one cat, but the cat-owning women were no more likely to develop brain cancer than their cat-free counterparts.
Yeah, absolutely right.
So this is cancer, and the other one is not cancer, it's suicide.
It's a behavior.
But,
Toxoplasma infection is known to induce behavioral changes, particularly, as we've mentioned before, in the ability of mice and rats to smell cats.
But in people, we've seen a few other articles over the past few months about the idea that most people are infected in the brain with not just T. gondii, but also neurocysticercosis. Right?
80% of France is positive for toxoplasma. 80%, Vince.
Seropositive.
80.
So, are you saying 80% of the French behave abnormally?
No, I'm saying just the opposite.
I think these studies are hard because there are probably confounding factors.
There are tons of confounding factors.
But I'm not qualified to really say whether they're correct or not. We should get someone who's works on this.
We should. Actually,
talk about it.
We've thought about this. A good friend of mine, John Boothroyd, would be happy to be a guest on our show sometime. He has expressed that.
Who is that
to me.
again?
John Boothroyd. He's at Stanford.
Okay. Spencer writes, Dear Vincent and Dickson, here's a news piece very relevant for TWiP, the first congenital case of Chagas disease in the U.S., with a great video from Dr. Jim McCarrow.
Oh, Jim. Yeah, he's out at UCSF.
You know Jim?
Yeah, I do, very well.
So he sends...
Studies metalloproteases.
What is the significance of a congenital case of Chagas disease, Dickson?
The significance, of course, is that that's just yet another route that you can acquire this infection from.
The boy was delivered by C-section.
Really? Oh, yeah, okay. No,
in August 2010. Enough. And then a few weeks later, it's in the second week after birth, his mother said she had Chagas. So they tested the child, and he turned out to be positive. So that means he acquired it from his mother.
But that's not a new finding. We've known about this.
But it's the first in the U.S.
That's first in the U.S. That's right.
And she had been infected in
Argentina,
Bolivia, but not the U.S.
No, of course.
But it's not going to spread to anyone else, is it?
We have to have the proper vector for this to happen, unless, of course, you're going to retain it if that was a female birth, right? And she then had children. Yeah, she could pass it on this way.
They gave him benidazole.
Benznidazole.
How dare they put a Z and an N together?
But they did.
His symptoms cleared up, and parasite test at 10 months was negative.
Really?
Mm-hmm.
He was lucky.
That drug is not approved in the U.S.,
No,
you know.
it isn't. I
it's
know
only
that.
available for investigational purposes.
It's a very toxic drug, Vince.
Really?
Yeah.
Okay.
Yeah.
Thank you, Spencer.
Yep.
Thanks, and keep up the great episodes.
We'll try.
All right, now we have another long letter, which I'm going to skip. We will save that till next time.
Okay.
Let's end here
Yes.
and say you can find TWiP on iTunes and at microworld.org/TWiP. And if you like TWiP, the best thing that you can do to help us is to subscribe over at iTunes. Or if you don't want to do that, just go to iTunes and give the show a rating of stars. You can give it as many as 5. And that helps us stay visible on the Apple iTunes directory. And that means we get more listeners. We don't get more money. We don't get any money.
No.
We get more listeners, and that's what we want, to spread the word and infect people with the parasite of TWiP and influence their behavior so that they work out more.
Some of them.
Which is good if people walk to work instead of taking the bus, right?
Yes.
We love to get your questions and comments.
We do.
Send them to twip@twiv.tv.
Yes.
And if you would like to find Dickson Despommier, he is at Trichinella.org.
Still.
And verticalfarm.com. Where
Still.
trying to solve the world's hunger problems. And medicalecology.com. Org. Very good, Dickson. Thank you for joining us today.
Oh, Vince, it's a pleasure as usual.
Yes. And did you enjoy discussing papers?
Loved it. Okay. Loved it.
You learned something, I presume.
A lot.
I did too.
I mean, a lot. Thank God for the internet. I'm not kidding.
It is a resource. Otherwise, we'd have to do this in the library.
Otherwise, we'd have to make lots of mistakes.
They'd kick us out, right?
It would be very slow.
I'm Vincent Racaniello, and you can find me at my blog, virology.ws. You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.